that's taking place in the female girl before she's even born. Okay, so there's a little bit of difference in the timing, but the genetic changes are still the same. So, we're going to look at a spermatogonium. And for those of you that may have forgotten from Bio 430, we'll look at mitosis first. Because mitosis continues to occur in the testis, but not in the ovary. Because there are no oogonia, the female is born with all of the oocytes she's going to have, approximately 300,000, a little bit plus or minus. So there's actually more that are produced, but some of them die before she's born. So there's no oogonia in the female. No mitosis occurs at puberty in the ovary, but it does in the male with the spermatogonium. So instead of drawing 23 pairs of chromosomes, a total of 46, which means in humans, N equals 23. A cell is considered to be 2N because you get 23 chromosomes in the egg and 23 in the sperm. And so chromosome 1 comes from the father, chromosome 1 comes from the mother, and they are considered to match as alleles. That gets too complicated to draw on the board, so I'm going to draw 2. Okay, so for this particular spermatogonium, N equals 2. So the mother contributed 2 chromosomes, and the father contributed 2 chromosomes. And one of the pair is an X and a Y. I don't care what the other one carries, but so this is a diploid cell. So it is a 2N, which means we have a total of 4 chromosomes. If N equals 2, and the mother gave 2, and the father gave 2 of this man, then we have a total of 4, all right? So we'll put the X as a red one, and the Y as a green. So the Y, and that's an X. And I don't care what the other one is, we'll put a blue and a black, all right? So that's our diploid cell, where here's our 1 pair, and here's our second pair, N equals 2, 1 and 2, okay? 1 from the mother, 1 from the father, 1 from the mother, 1 from the father. All right, so for mitosis, and I'm not going to keep drawing the cell or the nucleus, the cells replicate. If you recall, this occurs during the S phase of a cell cycle, and sister chromatids are formed. So the way they line up in metaphase is completely random. So those are sister chromatids. And then in anaphase, the microtubules shorten and pull them apart. So we end up having 2 complete cells that look like this. All right, so we would have 2 more spermatogonia. And they would have identical chromosomes. And this is mitosis. Okay? We have 2 identical appearing cells because they're 2 daughter cells that mitosis has occurred. And that's the purpose of mitosis, it's just to increase the number of cells. The gametes, the spermatogonia, the spermatocytes, or the oocytes are the only ones that undergo meiosis. Okay, other body cells do not undergo meiosis. So any cell in the body that I name, remember I said I would have a table or a chart, and I was going to have different cells, and I wanted you to give you their chromosomal pattern. So any cell that I name, that is anything beyond spermatogonium, primary spermatocyte, oocyte, secondary spermatocyte, a polar body if we're talking about the eggs, they're the only ones that are not going to be 2N. So a Sertoli cell is 2N. An interstitial cell, a cell Leydig making testosterone, is 2N. A muscle cell is 2N, or neuron is 2N, okay? The only thing that's not 2N is one of our developing gametes. So let's look at meiosis now, okay? So that's what spermatogenesis is. Remember, spermiogenesis was the change in appearance between spermatid and spermatozoan. So now we're going to look at meiosis, which is the change in the chromosomes. A little bit different. I guess I'll leave that up there for comparison. So we'll look at another spermatogonium. One of these spermatogonia that form will stay behind to undergo mitosis again. And the other one will go and form but it will become a primary spermatocyte. Okay, so we'll take one of these. And it's going to have the same chromosomes that it does down here. All right, and now it's going to become a primary spermatocyte. There is no cell division that occurs between a spermatogonium and a primary spermatocyte. The changes are chromosomal. So it's getting ready for its first meiotic division. So to do so, it's going to replicate. It's going to duplicate the chromosomes again. We're going to form sister chromatids, but they're going to stay attached to each other. So instead of having this pair separate, they're going to be together, all right, what we call a tetrad. So here's our replication of sister chromatids of the X. Here's our replication of sister chromatids of the Y. And so when they attach a pair from the mother and the father of the same chromosome, so again, now we have two X's, okay? And we have two Y's. The same thing is going to happen for our other set. Now I said this was from the mother and that was from the father, but they don't have to stay on the same side. This is totally random. So we can have the father's set of chromosome 2 over here and the mother's sister chromatids over here. Or they could all be the other direction. So these are called tetrads. So tetrads are a component of a primary spermatocyte. So I drew these a little far apart. I'll make my primary spermatocyte big, and it is bigger than the typical spermatogonium. Is it still a diploid cell? Yes, because we haven't done any division, so it's still going to be diploid. But because we have the duplicate copies still attached to each other, so it's still 2N, but this is the way I write it out. We now have each one copied and set together, so it's 2 parentheses 2N. Okay? So any primary spermatocyte or any primary oocyte, when we get to that, is going to be a 2 parentheses 2N. Okay? Now, I did mention that last week, but I did not talk about crossing over. Because these pairs of chromosomes are in close vicinity to each other, a piece of one chromosome will exchange with a piece of its